Hello and welcome to the Sleepy Scholar. You begin this evening's journey by entering a quiet space. The air feels hushed, like the silence of an ancient library after dusk. You notice how still everything seems, and yet beneath the calm, there is movement. Invisible rhythms continue, soft and hidden, steady as a heartbeat. This is not a library of stone walls and paper pages. It is smaller, infinitely smaller. It is inside you, within every single cell of your body. Each cell, so tiny it escapes your eyes, carries its own library. Not shelves of books, but strands of letters, twisted like ribbons. These ribbons are DNA, the language of life, a script more ancient than memory. You picture walking softly between rows of glowing shelves. The air is warm with quiet light, each strand of DNA shimmering like a candle. Every shelf holds instructions, written long before you were born. They hum like a faint song, reminding your body how to breathe, how to grow, how to heal. It feels as though you are stepping into the very heart of yourself. The library is vast, though each volume seems delicate. 4 letters form the entire language of DNA: A, T, C, and G. Simple at first glance, and yet arranged in endless combinations, they tell the story of every living thing. Like letters forming words, and words forming poetry, they carry rhythm and order. When you pause to imagine it, you see your body not as solid, but as a collection of instructions, carefully unfolding in time. Each of your cells, from the tip of your finger to the depth of your brain, carries this same library. There are trillions of them, and each cell keeps its own copy, faithfully preserved. The thought feels comforting, that within you, a pattern repeats itself, quietly ensuring that you remain you. The library of life has no librarians, no human hands to arrange its shelves. Instead, it is self-keeping, self-reading. Proteins, like gentle caretakers, open the strands of DNA, reading its letters line by line. Some build, some repair, some translate instructions into action. It is as if the books write themselves over and over, without ever tiring. You walk further down the candlelit aisles. The shelves stretch endlessly in both directions. Some letters are ancient, carried for millions of years, unchanged. Others are new, small variations that appeared like whispers in the text. Together, they form the story of your ancestry, not just human ancestry, but the ancestry of life itself. For a moment, you pause at one glowing volume. It feels as though you are touching history, though not the history of kings or wars. This is deeper, a history of survival, of adaptation, of tiny changes passed down like secrets. You realize that your body is not only yours, it is the result of countless lives before you, all adding their notes into the library you now carry. The spiraled letters are not random. They fold and loop, forming compact shapes so they can fit inside the tiny nucleus of the cell. You imagine whole encyclopedias condensed into a single bead of light. So much knowledge tucked into such a small space. It feels miraculous, though the library does not ask to be noticed. It simply does its work, quietly, faithfully, day after day. The library of life does not sleep. Even now, as you prepare to rest, your DNA continues its reading. It tells your cells to repair themselves, to knit proteins, to strengthen your body while you dream. You drift with the comfort that this hidden text works without effort from you. It is both intimate and anonymous, both personal and universal. You imagine yourself holding a strand of DNA in your hand. It stretches like a ribbon, fragile but endlessly long. If unwound, a single strand from one cell could reach for meters, and yet here it lies, folded with elegance, hidden away where your eyes cannot see. It is a reminder of how something invisible can hold infinite weight. The shelves in this inner library do not carry only your story. They carry echoes of everything that has ever lived. Because DNA is universal, shared by plants, animals, even the smallest microbes. Each uses the same 4 letters, though the sentences differ. You share part of your text with a tree, a bird, a whale. The differences lie not in the alphabet, but in the arrangement of its poetry. As you walk deeper into this quiet library, the air feels timeless. There are no clocks here, no rush. Life unfolds at its own pace, writing and rewriting itself slowly. You feel yourself slowing too, your breath falling into rhythm with the silence. You notice a corner where the shelves shimmer brighter. These are the regions of DNA that are read most often, instructions for life's daily needs. Other shelves remain dim, rarely touched, waiting perhaps for a time when they may be called upon. Like books in a library, some are borrowed every day while others gather dust until they are needed. The thought soothes you. Not every part of your story must be read at once. Some parts wait quietly, like hidden chapters, ready when the time is right. Life has patience built into its very core. In this calm space, you see how delicate the library is, and yet how resilient. Sometimes, a letter may change, a small slip, a mutation, and still the library continues, adjusting, adapting, carrying on. Errors may come, but life does not stop. It weaves even mistakes into the larger story. You pause again, your hand grazing the glowing shelves. There is something deeply human about this library, though it belongs to all creatures. It reminds you that order can emerge from simplicity. That meaning can arise from the smallest letters, endlessly repeated. The quiet brings your thoughts to rest. You begin to feel that you are not walking alone. Countless generations before, you walked with this same library inside them. Countless forms of life carry their own shelves, humming with the same alphabet. You are part of a chorus, your song joined with theirs. As you prepare to leave this vast inner library, you carry a new awareness. You know now that every breath, every movement is guided by this quiet script. It does not ask for recognition, only to be read faithfully, line by line. It is a comfort to know that inside you rests an order older than the stars, a code more enduring than stone. And yet, as you step toward the soft doorway of thought, a question lingers. If the library of life holds so many stories, where did it first learn to write? Who placed the first letters, the first notes of memory into its pages? You drift with that thought, and as the shelves fade from view, you begin to sense another story waiting. A story about how life learned to remember its enemies, how it began to leave small scribbles in its margins. You step again into the inner library of life, but now the shelves feel older. The glow of the DNA letters seems touched by history, as though the words have been revised many times before you arrived. You remember that every library carries not only its main texts, but also its scribbles, little notes in the margins, marks left behind by those who came before. And here, within the code of life, you find the same pattern. Long before you, before humans, before even animals or trees, there were simpler creatures. Their cells were small, without complexity, and yet within them lived a powerful need, the need to remember, to defend, to survive. Viruses, invisible threads of life on the edge of existence attack these cells. A virus, in its essence, is little more than a piece of code, an instruction trying to trick a cell into copying it. And so, the battle between virus and cell began, a story written long before memory itself. Imagine standing beside a single-celled bacterium, drifting in ancient waters. It has no eyes, no mouth, no awareness in the way you understand it. But when a virus enters, it feels the invasion, like an unwelcome hand tearing through the library shelves. The virus unrolls its own script, trying to replace the cell's poetry with its own. If successful, the bacterium becomes a factory for the virus, its library silenced, its shelves rewritten. But sometimes, the bacterium survives. It does something remarkable. It takes a small piece of the virus's script and tucks it into its own DNA. Not in the main text, but in a separate section, a margin, a notebook within the library. This is CRISPR, a record of past invaders stored like scribbled notes, so the cell can recognize the same intruder if it returns. You can almost see it happening. Each virus defeated, leaving behind a faint mark, a scar, a line of memory. Row by row, the bacterium collects these fragments. They do not control the main story, but they sit quietly in the margins, ready to be opened if danger comes again. It is as if life itself invents a diary, writing down every enemy it has ever faced. When a familiar virus returns, the bacterium does not read blindly. It opens the scribbled notes of CRISPR. It matches the letters, piece by piece, like comparing handwriting. And once it knows the intruder, it calls upon a special tool, a protein designed to cut. Cas9 is its name, and though you will meet it more fully later, here it first appears, like a sharp pen following the margins of the page. With its guidance, the bacterium can slice apart the virus's code before it spreads. You notice something soothing in this rhythm. Life remembers what harms it, then quietly protects itself the next time, like a tree scarred by a storm, or your own skin remembering sunlight. Cells, too, keep their memories, though in the language of DNA. Over time, these scribbles multiplied. Some bacteria carried long lists of enemies, each fragment a warning, each line a survival story. CRISPR was never a tool invented by humans. It was a gift of nature, refined over billions of years, hidden within the simplest organisms. All we did was notice it, read it, and learn how to guide it. You walk slowly down the aisles of this library, imagining each margin filled with faint, handwritten notes. Some lines are old and faded, the viruses they described long gone. Others are fresh, bright, with recent battle. It feels strangely comforting, this idea that even the smallest life keeps memory, keeps record, keeps story. You reflect on your own memories, how you keep journals, photographs, or unspoken recollections tucked away. CRISPR feels the same, not the main tale, but the background, the quiet defense. You realize that survival itself is a kind of poetry, written not in words, but in resilience. As you linger, you imagine what it must have been like for early scientists to discover this system. They looked into the DNA of bacteria and saw repeating patterns, curious clusters of letters. At first, they did not know what these repeats meant, only that they appeared again and again. It took time to understand that these were the scribbles of nature, the notes of past viral encounters. And when they realized it, they opened a door into a new kind of science. The library around you glows with a deeper light now. You see the strands not only as instructions for life, but as archives of memory, as defenses written long ago. It makes you wonder how much of life is shaped not just by what is written, but by what has been rewritten, corrected, and defended. Perhaps your own story, too, is made of scribbles, moments of pain, challenge, or loss that left marks in your margins. Yet those marks, like CRISPR notes, help you remember, help you protect, help you grow. The shelves stretch far, and you walk slowly, sensing the rhythm of survival written into every cell. What once seemed like random letters now feels like deliberate design, as though life carries not just instructions, but wisdom. You rest with the thought that life does not forget. It adapts, it remembers, it rewrites itself for the sake of tomorrow. And as the glow of this chapter dims, you begin to sense the next page waiting. If life keeps scribbles in its margins, then what tool does it use to act upon them? If the notes record danger, what hand holds the scissors that answer it? You drift with that thought, ready to meet the sharp, delicate instrument hidden in the spiral. You stand once more inside the vast library of life, its shelves lined with spirals of luminous code. The DNA you walked through earlier now glows faintly in the candlelit silence, a winding script etched in the language of possibility. Tonight, though, something new catches your attention. A sound, soft and deliberate, echoes through the stillness. Snip, snip. It is not the heavy clatter of scissors cutting paper, but the delicate sound of threads being carefully trimmed. You pause and listen. There, between the shelves of spiraled letters, a figure moves. It is not human, not alive in the way you are, but a tool designed by life itself. Cas9. You picture it like a pair of silver scissors, impossibly small, carried not in human hands, but guided by a strand of RNA. A messenger, a map, a lantern in the dark. Together they drift down the rows of DNA, searching for a precise spot, a specific sequence among billions. You imagine how impossible that search should be, yet somehow, with a grace that feels almost magical, the tool finds its mark. The scissors pause. They hover over the letters, glowing faintly like pearls on a string. Then snip, a clean, precise cut. Not careless, not destructive, but intentional. Like a gardener trimming one branch so that a tree can grow straighter, fuller, more alive. You walk closer, the quiet floor beneath you echoing softly. Around you, the DNA spirals breathe with possibility. For the first time, you realize that this library is not frozen. It is not static shelves filled with dust and age. It is alive. It can be rewritten. It can be edited. Cas9 feels like a librarian's assistant, walking with quiet purpose, carrying sharp but gentle scissors. It listens to the instructions given to it by a guide, RNA, a thread of information designed to match the sequence of letters it must find. Together, they are like a traveler and a compass. Without the compass, the traveler wanders endlessly. Without the traveler, the compass cannot move. But together, they arrive exactly where they are needed. And then, in a moment of pure precision, the scissors cut. You let yourself imagine this as a story unfolding inside your own cells. A mistake in the DNA, a letter out of place, can be trimmed away. A harmful sequence, one that carries the weight of illness, can be edited, replaced, healed. For centuries, humans looked at the script of life with awe, but with helplessness. The letters were there, written long before birth, unchangeable, unyielding. Now, with this tool, there is a doorway. the possibility of change. You picture a gardener in a moonlit orchard. Branches grow wild, some heavy with fruit, some twisted and tangled. The gardener moves with care, clipping only what must be clipped, reshaping the tree not to destroy it, but to give it balance. Cas9 is this gardener inside the living library, a guardian of precision, a tool of transformation. The sound of the snip echoes again, soft and rhythmic. Snip, snip. Each cut is followed by silence, as if the DNA itself pauses to take a breath, deciding how to respond. Because a cut is not the end. A cut opens space. And in that space, the cell itself chooses. It can repair. It can insert new letters. It can rejoin what was separated. You think about how remarkable that is. The scissors alone are not enough. It is the cooperation between the tool and the cell's own natural repair systems that allows change to occur. Like a sculptor who carves the first line of marble, but it is the stone itself that reveals the final form. Together, they create a balance of precision and possibility. You imagine walking past rows and rows of spirals, each one carrying the stories of countless generations. Somewhere in those spirals is the story of your eye color, your height, your ability to digest certain foods, your immune system's resilience. And somewhere, too, are the hidden flaws, small mistakes, invisible errors that might one day cause illness. Cas9 does not judge. It does not choose right or wrong. It simply listens to the guide it is given and follows faithfully. In this way, the scissors are neutral. They hold no morality of their own. Their sharpness can heal, but it can also harm if misused. That truth lingers in the library, like the faint shadow of candlelight flickering across the shelves. You feel both awe and caution. Tools, no matter how precise, always depend on the hands, or in this case, the minds, that wield them. Still, you focus on the beauty of the possibility. Imagine a child born with a disorder that steals away energy, trapping them inside fragile muscles. Imagine doctors using this tool to trim away the mistake in the DNA, restoring strength, offering freedom. Imagine fields of crops, resistant to drought and disease, growing strong with edits that help them thrive where before they would wither. The scissors whisper not just of correction, but of renewal. You pause before one shelf, where the spiraled letters glow brighter, as if inviting you closer. You see Cas9 drifting toward it, scissors poised, guide RNA leading the way. It stops at a very specific sequence, and with delicate care, it makes its cut. The strand separates, and in the silence that follows, you imagine new letters being gently placed into the open space. A sentence rewritten. A story changed. The thought soothes you that even in the heart of biology, change is possible. The library is not closed. It breathes with revision, with careful editing, with the possibility of new beginnings. And you, standing here in this vast hall of spirals, feel the weight of that hope. Your breathing slows as you follow the rhythm of the scissors. Snip. Silence. Snip. Silence. Like the pulse of waves against a shore. Gentle, steady, timeless. The sound lulls you deeper into the story of your own body, reminding you that beneath the surface of your thoughts, there is an orchestra of precision unfolding in silence. And yet, a question lingers in the soft shadows of the shelves. If DNA can be edited, if life itself can be rewritten with tools like these scissors, what does that mean for the future of the library. What stories might be changed, and what stories must remain untouched? The shelves fade into a softer glow, and you begin to notice something beyond the rows of spirals, a quiet hum, low and resonant, vibrating through the floor beneath you. It feels older than Cas9, older than DNA itself, a deeper rhythm, like the beating of a heart beneath stone. You pause, holding your breath, and wonder, perhaps the scissors are not the end of the story, but only a doorway, because beyond the shelves of letters, there is another force at work, older and more mysterious, something that hums beneath all life, waiting for you to listen. You sit at a desk, a blank sheet of paper before you. A pencil rests lightly between your fingers. You know the feeling, the beginning of a story. A letter takes shape, then another, and slowly words string themselves into sentences. But sometimes the lines stumble. A phrase is wrong, a word misplaced, a letter reversed. You pause, raise the eraser, and gently rub away the mistake. The smudge fades, and the paper opens once again, ready to be rewritten. This simple act of editing, so ordinary and familiar, has long been limited to ink and paper. Yet within your body, inside the cells that carry your story, the same principle now emerges. The genome, once thought permanent and unchanging, is revealed to be pliable, like parchment that can be revised. With CRISPR, scientists hold not only a pencil, but also an eraser, a pair of gentle scissors, and a brush of ink that can rewrite life's manuscript. Imagine your DNA as a library, its shelves reaching beyond sight. Each volume is filled with words, phrases, and instructions, written in a four-letter alphabet: A, C, G, T. Every letter has its place, every sentence its rhythm. But sometimes, a single letter falls out of order. One misplaced mark can alter a meaning entirely. Like a typographical error in a poem, it may twist beauty into confusion, shift harmony into discord. Now, with CRISPR, scientists can step into this library. They move quietly, not to burn or replace entire volumes, but to edit them carefully, word by word. A mistaken letter can be removed. A missing phrase can be added. A tangled sentence can be rewritten with grace. The genome is no longer stone, carved immutably by time. It is more like paper, able to be touched, corrected, softened into clarity. You may picture the process like a classroom exercise. The student writes, the teacher leans over, red pen in hand, circling errors, suggesting improvements. Yet here the teacher is nature itself, guided by the tool of CRISPR. Cas9, like a pair of scissors, locates the wrong sequence, snips it gently, and allows a correction to be written in its place. The process is deliberate and tender, as though the text itself were alive. Because it is. Pause a moment and imagine this editing in slow detail. The DNA strand, a spiral ribbon, floats before you. A hand reaches in, guided not by guesswork, but by precision. The scissors of Cas9 find the right spot, not any letter, but the letter that matters. A cut is made, neat and exact, and suddenly a window opens in the spiral. Into that gap, a new piece is slipped in, a corrected letter, amended phrase. The spiral closes again, seamless, as if the change had always been there. It feels almost like magic, yet it is science. A discovery born from years of observing bacteria, of noticing how they defend themselves against invading viruses. In their defense system, scientists recognized a tool, a way of storing fragments of viral DNA, a way of remembering, a way of cutting. What once was nature's memory of attack has become humanity's pencil of correction. The laboratory and the notebook merge into one continuous practice of writing, erasing, rewriting. Think of a child learning to write. Each attempt is imperfect, yet each revision brings the story closer to clarity. So too does CRISPR allow humanity to revise its biological story. A gene that once carried the heavy mark of disease can be rephrased. A mistake that caused suffering for generations can be gently corrected. The possibility of healing is written not in ink, but in the very alphabet of life. As you listen, let your breath slow to the rhythm of these edits. Each inhale feels like a new sentence arriving. Each exhale like a mistake leaving the page. The cycle of writing and erasing becomes the cycle of breathing, soft and natural. The genome, in this way, does not feel distant or abstract. It feels intimate, like a page inside you, slowly turning, slowly opening. Consider how profound this is. For centuries, humans believed the Book of Life was immutable, a sacred script written once and for all. To alter it seemed impossible, even dangerous. And yet now, through careful science, it is revealed to be something we can touch, something we can reshape with care. We are not replacing the book. We are learning to read it more deeply, to correct its misprints, to restore its lost words. You might wonder, what does it mean to hold such power? To erase suffering at the level of letters? To rewrite what was broken? These questions are not answered quickly. They linger, like the faint marks of an eraser, still visible beneath new writing. Every correction carries weight. Every choice opens a new path. Editing is never neutral. It bends the story in new directions. Imagine standing in a garden. Some plants grow strong, others falter. A gardener walks with gentle hands, pruning here, grafting there, ensuring that each stem has room to thrive. This is the feeling of CRISPR at work. Not a wild destruction, but a thoughtful tending. To cut carefully, to remove what hinders growth, to shape what can flourish, this is the gardener's art, and now also the biologist's. The metaphor continues to unfold. A manuscript is not perfect on its first draft. It takes time, patience, and revision. The genome, too, has drafts. Evolution has written across ages, leaving behind both poetry and error. CRISPR offers us a chance to participate in this long revision, to take up the pen alongside nature, to continue the writing of life itself. And yet, just as with your own writing, revision does not mean rewriting everything. Some lines carry beauty even in their imperfection. Some sentences are best left as they are. The art lies in knowing what to change and what to keep. In this, CRISPR is a reminder not only of science's power, but of restraint. To edit wisely is to edit gently. As you drift further into the rhythm of this thought, you feel the weight of the pencil in your hand again. You write a sentence, you notice its flaws, you erase, you rewrite. And in the act, you realize that the text itself is alive, flowing beneath your fingers, reshaping itself in dialogue with you. The genome is not a static monument, but a living manuscript, inviting collaboration. Softly, you return to the library of DNA. The shelves stand tall, the books glowing faintly. Scientists move between them, pens in hand, their steps slow, deliberate. They do not rush. Each correction is measured, each change weighed with care. In their patience, you see the rhythm of life itself, a rhythm that carries you towards sleep. As your eyes grow heavy, let yourself linger on one final image, a sentence rewritten with grace, a paragraph restored to harmony, a page smoothed by gentle hands. This is the gift of CRISPR, not only to change, but to heal, to refine, to bring clarity where once there was confusion. And as the ink dries, you sense that this is not the end of the story. Every manuscript, once revised, leads to another question, another page waiting to be turned. What then happens when editing moves beyond correction, when the story is not only restored, but reimagined? Imagine a story you have loved since childhood, a story that has traveled with you through the years. You know its rhythm, its images, the way the sentences fall like footsteps across a quiet road. But one evening, as you open the book again, you notice something you had not seen before. A word is misspelled. A letter lies out of place. It does not ruin the story, but it lingers. Each time your eyes brush across the page, the small mistake catches. It jars. The rhythm is broken. Now imagine that the story is not on paper at all, but written into the quiet folds of your body. The script is not black ink on white sheets, but a sequence of molecules spiraling inside your cells. This is your genome, the manuscript of life, written in 4 letters, A, C, G, and T. For most people, the words align in harmony, forming the long sentences that guide every breath, every heartbeat, every gesture. But for some, a single letter has slipped. A word is fractured. A sentence bends toward pain. Genetic diseases are these broken sentences. A child is born with a word misplaced in the story of their blood, or muscles, or vision. The story does not stop, but its music is altered. Hemophilia is a sentence where the ink for clotting has been smudged. Cystic fibrosis is a passage where a single missing piece of code turns the lungs into pages that stick and stiffen. Sickle cell disease is a line reshaped, folding blood cells into crescents that struggle to flow. Each is a reminder of how fragile language can be, how much depends on the smallest letter. For centuries, people lived with these stories written in permanence. Doctors could soothe symptoms, but the text itself seemed untouchable. The genome appeared like stone, carved and unyielding. You could not erase, you could not rewrite. The body carried its sentences faithfully, whether perfect or painful. Then, in the stillness of laboratories, a new idea emerged. What if the genome was less like stone and more like a manuscript? What if its words, though hidden deep in the nucleus of a cell, could be read, erased, even rewritten? What if the painful typo could be healed at its source? This is where CRISPR steps in, not as a heavy chisel, but as a delicate pen. The letters stand waiting, and CRISPR brings a tool that can find the exact place where the mistake lies. It does not guess. It does not wander blindly. It knows the coordinates, like a reader flipping to a particular page, scanning for a particular line. Once it arrives, it does something astonishing. It makes a cut, sharp, precise, but not reckless. The DNA is opened, as if the page has been lifted from the binding, and then a correction can be placed. A misspelled word is restored, a broken sentence repaired. Pause for a moment here, and notice the gentleness of this act. Life itself, rewritten in quiet strokes. The hope it carries is not abstract. It is felt in the breath of a child who can now run without gasping, in the strength of muscles that once weakened too soon, in the blood that flows smooth instead of jagged. Take, for instance, sickle cell disease. For generations, families carried this word error, passed silently from parent to child. The red blood cells, bent into sickles, could not glide easily through veins. They clumped, they tore, they brought waves of pain. Hospitals became second homes, and futures felt narrowed. But with CRISPR, scientists found a way to lift the word back into place. They entered the cells where the mistake lay, guided CRISPR to the precise line, and offered a new, corrected version of the sentence. The cells began to write themselves differently. Blood flowed with new rhythm. The story of the body shifted from suffering towards strength. This is not just science, it is poetry living in flesh. It is the idea that pain carried since birth may not be a destiny, that a sentence once thought permanent can be softened, healed, made whole. But healing does not only mean repair. Sometimes it means protection. CRISPR can act like an editor who not only fixes mistakes, but shields the manuscript from future harm. In experiments, scientists have used it to disarm viruses, removing their ability to weave chaos into our cells. Imagine a page where invasive ink threatens to smear across the lines. CRISPR steps in, not with violence, but with precision, removing the intrusion so the story can continue in clarity. Still, as you listen, let the gentleness of caution rise alongside hope. Editing the book of life is powerful, but power requires care. If you have ever tried to erase on paper, you know the smudge that can remain. In the genome, even a small, unintended change could echo. This is why scientists move slowly, deliberately, as though walking barefoot across glass. They test, they question, they return again and again to ensure that healing truly heals, that correction does not create new mistakes. In the quiet of these reflections, you may notice something larger stirring. CRISPR does not only offer the chance to heal disease, it reminds us of the nature of life itself, that it is written, yes, but not locked, that our bodies are stories in progress, manuscripts still open to revision. Where once we thought of fate, we now think of possibility. Where once we thought of permanence, we now think of change. And in this recognition, there is a deep calm. For just as you drift towards sleep tonight, your own cells continue their quiet work. Repair enzymes mend small breaks. Proteins fold and refold. Life edits itself continuously, not waiting for laboratories, but carrying on in silence. CRISPR is only an extension of what life already knows, to read, to notice, to mend. So you rest in the awareness of possibility. You rest in the vision of broken sentences becoming whole again. And you rest in the thought that stories, even the most painful ones, may yet find new endings. Let that thought linger. Let it settle like a bookmark, slipped gently between pages. For as healing unfolds in the body, another question arises quietly at the edge of your awareness. If we can edit the sentences of life, what else might we shape, not in the body alone, but in the future of entire species? The page does not answer yet. It waits open for the next chapter. Close your eyes and imagine the library of life. It is vast, stretching further than your mind can see, shelves upon shelves, filled with books that hum softly with the rhythm of living. Each book is a species, each page a strand of DNA, each letter a base. Adenine, thymine, cytosine, guanine, arranged in sentences older than time. And though you are only one small reader in this library, the script inside you is shared, echoed, mirrored across countless forms of life. The story of DNA does not belong only to you. It threads itself through wheat fields bending in the wind, through rice paddies shimmering with the sun's reflection, through tomatoes ripening slowly under a warm sky. The same alphabet that gives rise to your eyes also gives rise to the green chlorophyll of a leaf. Life feels like a chorus, all voices singing with the same few notes, arranged in endless ways. CRISPR, the quiet editor you met before, does not stop at human sentences. Its pen is carried into the fields, into barns, into laboratories where seeds wait for their first breath of soil. Imagine a scientist kneeling at the edge of a field. In her hands, she carries not only tools of metal and glass, but also the possibility of shaping a harvest. She holds seeds that have been gently touched by CRISPR's guiding hand. These seeds contain the same words as their ancestors, but some of the lines have been softened, corrected, made more resilient. Perhaps a single letter in their script has been shifted, so that when the summer sun burns too harsh, the plant can still keep breathing. Perhaps another letter has been added, so the stalk resists the insects that once feasted on it. Each change is small, like nudging a comma or smoothing a phrase, but in the field it becomes an orchestra of survival. You picture the plants rising together, millions of blades of green lifting toward the sky. Each one carries a slightly altered sentence, one that has been designed not for beauty on a page, but for strength in the soil. In their presence, you can almost hear the hum of a future where hunger does not press so hard against human lives. There is a gentleness to this vision, but also a gravity. Food is not simply nourishment. It is memory, culture, ritual. A loaf of bread is more than wheat. It is the smell of a kitchen, the warmth of a shared meal, the history of hands kneading dough for generations. To alter the script of crops is to touch not only biology, but also story. And so each CRISPR edit in the field must carry both humility and care. Still, the possibilities unfold like ripples on water. Imagine rice that carries extra nutrients, grains golden with vitamin A, feeding children whose bodies once knew only deficiency. Imagine corn that can grow in dry, cracked soil where rain no longer lingers, its resilience echoing hope in places once thought barren. Imagine bananas resistant to the blight that once threatened to erase them, preserved for the sweetness of future generations. You see, CRISPR does not create entirely new books in the library of life. It does not invent languages never spoken. Instead, it refines what is already there, returning to familiar pages and adjusting a word so the sentence holds. This editing is subtle, often invisible to the eye, but it carries the potential to shift the arc of survival, and the story moves beyond plants. Walk with your mind into a barn, where animals breathe softly in their stalls. Cows stand in a row, their flanks rising and falling with steady calm. Here, too, the editor's touch is present. Perhaps an edit helps a cow resist a virus that once spread silently among her herd. Perhaps pigs are given resilience against a disease that caused farmers and families deep loss. Each edit whispers not only of science, but of mercy, a way of lessening suffering, of giving creatures a gentler path through life. Even mosquitoes are not left untouched by these pages. You imagine a lab where their tiny wings buzz against glass. With CRISPR, some are edited so they no longer carry the parasite of malaria. In time, whole populations could shift, their dangerous cargo erased, their flight becoming only the sound of an insect. not the herald of disease. The thought feels like opening a window in a fevered room, letting cool air drift in. But as you drift deeper into this vision, you feel the weight of questions rising. If we hold the pen, how do we decide what lines to change? Who chooses which hunger to erase? Which creature to save? Which possibility to pursue? The library of life is vast, and though our hands now carry the editor's tool, the stories are not ours alone to write. You pause here, letting the uncertainty settle like dust in sunlight. It is not an unwelcome feeling. It reminds you that power must be balanced by humility. Every time CRISPR touches a page beyond human bodies, it touches ecosystems, relationships, cycles of life that extend beyond what we can always see. The Earth's text is delicate. Even when we edit with care, the echoes travel further than we know. And yet, hope lingers. It lingers in the face of children no longer hungry. It lingers in the sight of forests not stripped by disease. It lingers in the hum of bees visiting blossoms, carrying the promise of harvest forward. CRISPR is not a guarantee, but it is a possibility, an opening in the story, a space where resilience might be written in new ways. You sense the rhythm of this moment, the shared nature of DNA, the shared nature of survival. You and a blade of grass, a sparrow, a salmon swimming upstream, all are readers of the same alphabet. The thought is humbling and also comforting. Life, in all its forms, is a conversation written with 4 letters, rearranged in endless songs. CRISPR is a hand that joins that conversation, carefully offering edits, hoping to help the voices continue. As you sit with this, the image of the library of life returns. You walk through its halls slowly, fingers tracing the spines of books that breathe with unseen energy. You pause at the shelves marked for plants, for animals, for microbes, invisible yet essential. Each book hums with its own story, but the letters are familiar, the script continuous. You realize that the library is not separate from you. You are inside it, written into its pages, even as you read. You close your eyes more deeply now, and you feel the library dissolve into sensation. The scent of soil after rain, the warmth of sunlight on your arm, the sound of leaves shifting in a breeze. All of these are chapters of the same story. And somewhere, in the silence, CRISPR continues to work like a quiet editor, not rewriting the book of life, but gently, carefully, repairing, strengthening, preserving. The earth breathes with these changes. You imagine standing in a field at twilight, watching fireflies flicker above the crops, their lights echoing stars just beginning to appear. The plants sway gently, carrying new strength in their stems. The animals rest more easily in their barns. The air feels full of potential, like a story that is still being written, its next chapter not yet known. And here, with this image, you pause. You let yourself breathe slowly, holding the thought that DNA connects you to all living things, that the edits written in one corner of the earth ripple into another, that science and story are never separate, but threads of the same woven cloth. The library is still open, its shelves endless. CRISPR's pen still rests in careful hands. The question lingers like a page not yet turned. If life is a shared text, then what new sentences might we dare to write together? And as you rest in that question, your thoughts drift toward another horizon. What happens when the pen moves not only across pages of survival, but also across the boundaries of what life itself can become? You drift further into the library of life, and the shelves stretch longer than the horizon. The air is still, yet heavy with questions. Every book around you carries a story of survival, of inheritance, of the delicate way time weaves its hands through generations. Some pages have smudges, others are missing words. Some are clear, precise, and strong. As you move, you realize that every mark holds meaning, and to alter even a single one is not just a task of science, but of conscience. CRISPR, with its sharp precision, offered humanity a key to edit these books. But with that key comes a door, and with every door, a decision of whether to open it or let it remain closed. You pause at the thought. The stillness of this moment stretches, like ripples forming in a quiet pond. You lean into the thought of possibility. What if disease no longer carried the power it once did? What if a child could be freed from suffering before their first breath? The promise of relief hums gently in your mind, like a lullaby of hope. You feel the warmth of this vision, healthier lives, lighter burdens, the chance for generations to step into the world unshackled. But then another wave follows. What happens when the pen does not stop at healing, but begins to wander toward preference? A taller frame, a sharper mind, a particular shade of eyes. You sense the unease beneath these choices, like altering the rhythm of a song that was once whole in its imperfection. You wonder what is gained and what might be lost. The questions gather slowly, like dusk. They do not press harshly, but they do not leave. If we can change the script of life, who decides which chapters deserve editing? Whose hands should hold the pen? Scientists, parents, governments? Or should the book remain untouched, its typos and stains cherished as signs of being human? You walk through this library, and the shelves begin to whisper. They remind you that every line of DNA is more than code. It is memory. It carries the storms survived, the climates adapted to, the risks endured. The so-called errors may be, in hidden ways, the very ink that allowed existence to continue. A flaw in one place, a strength in another. The candle of thought flickers softly in your hands. You remember that in science, clarity often comes dressed in shadows. The right path is rarely straight. To edit is power, but power bends with intention, and intention bends with values. You breathe into this awareness, letting its weight settle, not as fear, but as humility. Imagine for a moment a field of wheat. CRISPR makes it strong, resilient against drought and disease. Hunger shrinks, families eat, communities thrive. Yet, in another corner of the world, a different hand edits a different seed, not for survival, but for control, for profit, for gain. The same tool, but two stories. You realize then that science itself is not the author, it is the ink. Humanity chooses the sentences. The thought expands, filling you with quiet awe and unease together. There is beauty in the power to heal, but also danger in the desire to perfect. Perhaps perfection is not the aim at all, but balance, the balance between what can be fixed and what must be accepted. The balance between rewriting life and respecting its original voice. You pause in the corridor of shelves. A single book rests open on a wooden table. Its pages glimmer faintly, as though alive. You lean closer, and you see a passage that reads, "To change is to care, to refrain is to care. The heart of the question is not the ability, but the reason." The words sink into you, gentle and steady. They remind you that every choice, whether to edit or not, is a reflection of who we are and who we wish to become. Your breath slows as you trace the edges of this thought. You sense how the future of CRISPR is not just scientific, but profoundly human. It is a mirror, reflecting our hopes, our fears, our vision of fairness, our definition of what it means to live well. For some, the questions may carry urgency. A family faced with a genetic illness feels the pull toward change with all the gravity of love. For others, the questions may carry restraint, a belief that life must be lived as it unfolds, untouched by human hands. Neither answer is simple, neither path free of consequence. You let the thought expand, like smoke rising slowly into night air. Perhaps the greatest wisdom lies not in having answers, but in holding the questions with care, to acknowledge them without rushing, to cradle their weight gently, as one cradles a sleeping child. The questions themselves shape us, reminding us that knowledge alone is not enough. Compassion must walk beside it. The library grows quiet again. You stand between the rows of books, surrounded by millions of silent letters, each waiting, each alive. And you realize the question is not only what can we change, but what do we choose to protect? Some words on these pages are not ours to rewrite, no matter how tempting the ink in our hand may feel. The shelves begin to fade. The wooden floor softens under your feet, and you sense the world of dreams drawing nearer. Yet the echo of the questions remains, flowing gently within you. Every choice, every edit, every restraint is part of the unfolding narrative. The story of life is not only written in genes, it is written in the values we carry as we read and rewrite. And as the last candle dims, you feel yourself leaning into a new thought, one that is less about editing and more about listening. What if the genome is not only a text to alter, but a voice to hear? What if, within its lines, it whispers truths about our connection to all living things, truths that wait for us to listen before we decide to write again? And so you drift, carrying the soft weight of these questions. In the quiet between pages, a new wonder rises, not about what you can change, but about what the genome itself might be trying to say, if only you listen closely enough. You lie in stillness, your breath slow, your body heavy, yet your mind remains open, drifting gently like a leaf on a calm river. The world within you begins to shimmer again, as though invisible musicians gather in the folds of your cells. Tonight, they play not a perfect melody, but something more alive, something unpredictable, the sound of chance itself. Every living thing carries music within its genes. The letters of DNA are like notes on a page, arranged into patterns, rhythms, harmonies. Most of the time, they repeat familiar themes, creating reliable songs that hold life steady. But sometimes, a note shifts, a letter changes, a phrase is added or removed. These are mutations. At first, they may feel like errors in a song you know by heart, like a violin string struck too hard, or a drum beat landing just off rhythm. But listen closer. Not every shift is a mistake. Some are beginnings of entirely new melodies. You imagine yourself inside a vast hall of sound. Each wall hums with a different piece of music, some quiet, some dissonant, some bright as sunlight breaking through clouds. Mutations are not single notes, but the force that lets the music evolve. Without them, every creature, every tree, every cell would remain trapped in repetition, a loop of sameness without surprise. It is variation that gives life its resilience. It is change that writes new verses for the song of existence. In the softness of your drifting thoughts, you see a child humming a tune never sung before. That is what a beneficial mutation can be, a gift to the future, passed quietly through the generations. The hemoglobin in your blood, the shape of your immune system, the colors in your eyes, each carries a history of countless mutations, some fragile, some powerful, all shaping you into what you are. But not all mutations bring harmony. Some throw the music into disarray, a sudden silence where notes should be, a distortion so strong the melody struggles to continue. Cancer, inherited disorders, weakened functions, these are mutations too. They remind you that chance is both beautiful and dangerous, both creator and destroyer. And yet, as you rest here, listening to the echo of your own heartbeat, you realize this balance is the price of creativity. Without risk, there is no new music at all. Scientists step into this hall of sound with new tools. CRISPR, precise as a conductor's baton, allows them to choose which notes remain, which are silenced, which are rewritten. With it, a dissonant chord of disease can sometimes be softened into harmony. A single mutation that brings suffering can be corrected, as if erasing a sharp edge from an otherwise graceful song. The dream of healing grows closer with each discovery. But you sense the tension too. If every flawed note is corrected, what happens to the unexpected beauty hidden within imperfection? Would the symphony of life become too controlled, too narrow, losing the surprises that variation brings? A butterfly's wing pattern, a bird's call, a human's unique face, all rise from chance meetings of mutations, most never planned. To silence them is to risk silencing the creativity of nature itself. You recall the wind playing through chimes outside a quiet house at night. Each gust creates a slightly different tune, none repeated, none predictable. The randomness is what makes the music endlessly alive. Mutations are like those chimes, ringing with possibility. CRISPR may allow us to still the wind, to control the sound, but should every breeze be tamed? Should every chance be rewritten? Your body relaxes deeper into the rhythm of this thought. You see a spiral staircase of DNA glowing before you, each step shimmering like glass. Some steps are solid and familiar, others flicker, uncertain, reshaping themselves as you watch. These flickers are mutations, altering the way forward. Sometimes they break, forcing a harder climb. Sometimes they form bridges, opening paths that never existed before. Every creature who ever lived walked this staircase, guided not by perfection, but by change. Life's history itself is a record of mutations that endured. The fins of ancient fish shifting into the legs of creatures who walked on land. The voice box of primates transforming until humans could sing. The pigments that allowed flowers to call insects with color. Each step came from a note that was once strange, once out of place, but over time folded into harmony. Evolution is not a march of flawless design, but a song composed of endless variations, most forgotten, a few remembered in the bodies of the living. Even now, as you breathe, mutations are happening quietly within you. Cells divide, letters are copied, and sometimes the copying slips. Your body carries guardians, enzymes that repair, systems that check for errors. Most changes are corrected before they can take root, but some slip through, like whispered notes in the background of the song. You will never know most of them. They vanish without trace. A few linger, And in the vast ocean of time, one of those whispers might change everything for those who come after you. This realization brings you to stillness. The future is not set in stone, but is being written even now in tiny changes hidden from sight. You rest in awe of how delicate and vast this process is. Every life is both a repetition of old songs and a rehearsal for new ones. You are not only a listener in this concert hall, you are part of the music itself. The question lingers: how much should we, as humans with new tools, intervene in this concert? Should we tune every note to match our sense of beauty, or let chance compose some of the melody? The decision is not simple. Healing is mercy. Control can be safety, but too much control may silence the symphony of surprise that made life what it is. The music continues around you, soft and endless. You imagine yourself lying in a field at night, the sky overhead pulsing with unseen waves of sound. Stars become notes, galaxies become chords, mutations ripple like improvisations in this cosmic score, small changes that build towards something larger. You cannot predict where the song will go. That is its wonder. That is its gift. And so you let the thought rest in you, like a final note held long and gentle. Every error, every shift, every variation is part of a greater whole. Even the fragile, even the broken, belong in the music of existence. To listen is to accept both harmony and dissonance, both chance and choice. Your breath slows further. The hall fades, yet the hum remains. The music of mutations continues, patient and unending, waiting for you to lean closer, waiting for you to hear what comes next. You wonder quietly, if chance writes the songs of life, who holds the silence between notes? In that pause, a new mystery waits, one that drifts like a shadow into the next chapter. You close your eyes and imagine a book with no final page. Its cover is soft, unfinished, and its spine stretches into the distance, too far to see. When you lean closer, you realize the words are not written in ink, but in sequences of 4 repeating letters, arranged like patterns of music. Each letter stands for a base, each base for a possibility, each possibility for a life that could unfold. You breathe slowly, sensing the weight of that idea, a manuscript that is not just about you, but about tomorrow itself. The boundless manuscript of tomorrow is written in DNA, but unlike any ordinary book, it is not static. It is alive, whispering with every breath, every heartbeat, every new cell that divides inside you. When you think of the future, you often picture things made by human hands, cities, machines, inventions. Yet the truest manuscript of the future is woven deep inside the living code, quietly shaping not just how you live, but how every being after you might come to live. Pause for a moment. Imagine holding a pen that could reach into this manuscript, not just to copy or preserve, but to change. To add a word here, remove a word there, even to rewrite an entire sentence of existence. That is what CRISPR offers, the ability to edit life itself, to alter the script that stretches beyond your own story and flows into generations yet unborn. You may wonder, what does it mean to edit the future? In simple terms, CRISPR is like a pair of molecular scissors guided by a map. With astonishing precision, it can find a single word in the immense encyclopedia of DNA and change it. Correct a misspelling, insert a new phrase, or silence something harmful. Scientists first noticed this system not in humans, but in bacteria, who used it as a memory bank to defend against viruses. What was once nature's shield is now being reshaped into humanity's tool. You picture the act of editing as delicate as turning a page in silence. Scientists guide CRISPR like librarians tending to fragile manuscripts. But this manuscript is not made of paper. It is living, breathing, branching forward into futures unseen. One small change today ripples outward, not just through the body it touches, but through the descendants that may follow. Consider a family tree. At its roots are people of long ago, and at its branches are those not yet born. When CRISPR edits a single seed in this tree, the branches change shape. Some diseases that once haunted a family may never bloom again. Certain risks may vanish, like shadows erased before they fall. Yet with every edit comes a question. What else might shift in ways we cannot predict? The thought is both comforting and unsettling. On one side, you see relief from suffering, a child spared from a painful illness, a life free from burdens that once seemed inevitable. On the other, you sense the unknown melodies that may be silenced before they can be heard, the surprises of evolution, muted by choices made too soon. You breathe deeper. Your mind drifts into metaphor. Imagine the manuscript of life as a symphony performed across endless generations. Mutations are like improvisations, unexpected notes that sometimes add harmony, sometimes discord. CRISPR is like a conductor stepping in, choosing which notes to keep and which to quiet. The performance grows more intentional, but perhaps also less wild. And in that thought lies the question, is a future more perfect always better, or is it the imperfections that make it beautiful? Let yourself rest in the image of a child yet unborn. Their story has not begun, yet pieces of it are already written in the spirals of DNA they will inherit. Perhaps their eyes, their height, their health, perhaps even small tendencies of personality. If CRISPR can edit these before the child takes their first breath, what does it mean for the story of tomorrow? Is the child more theirs or more ours? Science is not destiny, but it is possibility. Today, CRISPR is still a young tool, most often used in laboratories or in carefully monitored treatments. Yet the dreams around it stretch vast, curing genetic disorders, strengthening crops against famine, even editing species to resist extinction. The manuscript of tomorrow could become a patchwork of choices made today. You pause, imagining the scale. It is like standing in a vast library, every shelf filled with books not yet written. Each edit, each decision, determines which books will be added, which will never appear, and which will change their endings entirely. The sheer weight of possibility makes you exhale slowly, as though to steady yourself against the immensity of it. And still, you wonder, who should hold the pen? Should it be scientists, carefully weighing risks and benefits? Should it be societies, voting on what futures are worth creating? Or should the manuscript be left mostly untouched, guided by the slow hand of evolution as it has always been? There is no single answer, only the gentle pull of a question that stretches across time. As your mind drifts deeper, the manuscript becomes less a book and more a living river. You see words flowing like water, carrying meanings downstream. Each drop touches another, joining to form currents of inheritance and change. If you place your hand in the river, you may shift its flow. But the river is endless, its path winding into valleys and oceans you cannot yet see. Editing today is like casting a stone. The ripples move outward, beyond sight, beyond prediction. And yet, in this stillness, there is also wonder. Perhaps the manuscript of tomorrow is not meant to be perfect, but expansive, a place where science and chance, intention and accident all weave together. You sense that to edit is not only to control, but also to participate, to be co-authors in a story that began long before you and will continue long after. Your breathing slows with the thought. The room around you feels quiet, like the hush of a library at night. You can almost hear the rustle of pages turning in that endless book. The letters shift, align, and dance. Not just words, but possibilities. Somewhere in those possibilities lies a future where illness is eased. Somewhere lies a future where curiosity carries us further. And somewhere lies a future we cannot yet imagine, waiting like a blank page for tomorrow's ink. You drift with the awareness that time itself is a kind of author. Even without human hands, the manuscript keeps writing. Generations come and go. Mutations arise and fade. Life bends, adapts, experiments. CRISPR is simply one more hand at the table, one more brushstroke on a canvas that will never be finished. The manuscript of tomorrow is boundless, not because it has no limits, but because it always invites more. More edits, more questions, more possibilities. You rest in that invitation, knowing you do not need to answer everything tonight. You need only to wonder, to let your curiosity breathe as softly as your body does now. And as the manuscript stretches beyond your sight, you sense that its next pages are not only about biology or science. They are about meaning, about the ways in which humans choose to shape not just their bodies, but their worlds. The science of editing DNA is, in the end, also the science of asking what kind of future feels worth writing. Your eyelids grow heavier. The manuscript fades into the horizon, but its presence lingers, a reminder that every future is a story still unfolding, waiting for someone to turn the page. And as you rest with that thought, you wonder, if tomorrow's manuscript can be shaped by our hands, then perhaps the deeper question is not how we edit, but how we choose. And that question opens softly into the chapters yet to come. The library has been alive with whispers, pages shifting in the breeze of discovery. Tonight, as you step deeper into its halls, a stillness settles. The echo of footsteps fades, leaving behind a silence that feels like velvet, draped across the shelves. You reach for the familiar tome, the one you have been reading through the chapters of life itself. Its cover feels warm, as though it has absorbed every story told within. This is the Book of DNA, the ancient manuscript that carries every line, every correction, every possibility. And now, at this hour, it feels time to close it. Closing a book is never an erasure. It is an act of pause, of rest. The words do not vanish. They wait quietly in the dark, ready to be remembered, ready to be continued. So too with the language of life. CRISPR, with its delicate scissors and ink, has not ended the tale. It has only written in margins we never knew existed. And when you close this book now, you are not turning away from its meaning. You are resting your hands upon its cover, feeling its quiet weight, letting it breathe in the dark. As you breathe, you notice your chest rising and falling with a rhythm older than thought. Each inhale feels like a sentence beginning. Each exhale, a gentle punctuation. Your body itself is a manuscript, written line by line in cells that renew, in proteins that fold and refold. You are both reader and story. To close the book of DNA is not to walk away, but to listen more deeply to the story unfolding inside you. Imagine, for a moment, a library that exists not in wood or stone, but in the quiet chemistry of your being. Millions of books, each one tucked into the heart of a cell, waiting with patient endurance. These volumes never sleep, even as you do. They hum and work, repairing what frays, copying what fades. Their letters do not blur when you close your eyes at night. They keep writing, faithful to their duty, shaping the silence into continuity. The act of closing the book tonight is not about shutting out light, but about softening its glow. CRISPR is still there, whispering in the background, like a lantern left burning at the far end of the hall. Its promise stretches outward, into tomorrow, into generations you may never see. But your place now is here, at the edge of this quiet, where curiosity gives way to rest. You recall how the manuscript has shifted through time, mutations weaving missteps into hidden poetry, evolution turning accidents into adaptations. Life has always been a restless editor, crossing out, rephrasing, experimenting with endless drafts. CRISPR is not so different. It is our hands guided into the margins, making notes beside the author's lines. But unlike the hurried scribbles of human correction, these edits carry gravity. They ripple outward, influencing not only today, but the tomorrows stacked high on shelves we cannot yet climb. So you close the book gently with reverence, not to lock it away, but to let its lessons settle into your marrow. There is wisdom in resting, in not racing to read the next chapter too quickly. Books, like bodies, need time to digest. The cover presses against your palms as though reminding you that knowledge is not just absorbed but lived. Outside the library, night deepens. The stars above you appear like faint letters, scattered on a page too vast to bind. Each one shines with the quiet persistence of DNA, repeating its signal across light years, rewriting itself in the dark. If DNA is the script of life on Earth, perhaps the cosmos carries its own manuscripts, still unread, waiting for another reader, another dreamer, another dawn. And here you are, a single reader closing one book, while infinite shelves remain. The thought is not overwhelming, but comforting. You do not need to hold all stories at once. Tonight, you need only rest in the knowing that one story, the story of CRISPR and the story of life, has been gently set aside, its glow intact, its promise unwritten. As you breathe deeper, you imagine your mind as another page turning, another line softening. Sleep itself is a kind of editing, a nightly revision where unnecessary noise is struck out and memories are stitched carefully into the longer manuscript of who you are. Neurons rearrange, proteins adjust, dreams flutter across the page like footnotes in the margins. Closing your eyes is not an end, but an invitation to another kind of reading, where the book continues in silence. The gentle closing of the book teaches you something about endings. They are never truly endings. They are doorways, thresholds into pauses that prepare you for what follows. Even as the pages fall quiet, the story has momentum. Life is rewriting itself with or without your awareness, carrying on the same work that has persisted for billions of years. You are part of that work, even as you rest. You linger on the thought that all things are manuscripts in progress. Every cell, every tree, every drifting star is a draft in motion, revised endlessly by forces too vast to name. To live is to be a paragraph rewritten again and again, sometimes boldly, sometimes gently. And if you ever feel incomplete, it is only because your story is still unfolding, still carrying forward into chapters yet unformed. The library waits patiently. The shelves do not collapse when you walk away. The book does not mourn when you close its covers. It knows, as you now know, that stories need rest as much as they need telling. To pause is to honor the weight of what has been read. You place the book back upon the table, its cover gleaming faintly under the low light. The silence deepens around you, and in that silence, you hear a truth more profound than words. The story is never finished. It flows like a river beneath the surface, carrying whispers into tomorrow. And as you step away, you feel your breath slow, your body soften, your mind drift. The book is closed, yet the manuscript continues. You are carried by its current, held by its promise, soothed by its continuity. Perhaps the real manuscript is not the one bound in pages or hidden in cells, but the one unfolding in you now, the gentle, unfinished story of being alive. And as that thought lingers, you feel the edges blur, the sentences dissolve, and the next question rise like a faint glow on the horizon. What other stories are waiting, not in books, but in dreams?